Welcome to the Center for Nursing Research Lab at the CSIC School of Nursing. I'm Sharvari Kamath. I'm the Senior Research Associate and I manage the lab here. This lab was made to provide flexibility for the student and faculty research needs here. We have multiple portable workbenches. We have a tissue culture room where we have 2 CO2 incubator units, a refrigerated centrifuge and 2 biological safety cabinets for sterile applications. We have 2 biological safety rooms which have chemical fume hoods. We have a freezer area where we have -80°C freezers and which are connected to a alarm system. And we also have a Millicue water synthesis for our ultra pure water needs. In addition to the standard laboratory equipment and supplies, we also have a Perkin-Elmer Victor plate reader which we use for ELISA, a Luminex 200 system which we use for our multiplex needs, a Thermo Fisher Ultimate 3000 HPLC system, Applied Biosystems equipment for real-time PCR. We also have a traditional PCR system and we have a Piccolo chemistry analyzer. ELISA is the most popular assay that we use in our lab. So ELISA stands for Enzyme Link Immunosorbent Assay, and it's a plate-based assay technique which is used for detecting soluble substances such as proteins, peptides, antibodies and hormones. And the most crucial element of ELISA is the highly specific antibody-antigen interaction. In the CNR lab, we use the Victor plate reader for our ELISA assays. It's a multi-mode plate reader which detects absorbance, luminescence, fluorescence intensity, time-resolved fluorescence and fluorescence polarization. It is great for everyday biochemical assays and including kinetic measurements, and it includes features such as temperature control up to 65°C, a shaker for shaking the wells in between the plate reads, and it comes with an analysis software. The Luminex Multiplex System 200 is used for multiplex assays. Multiplex assays give users flexibility for multiple analyte detection. Now, ELISA can accurately detect and quantify individual proteins with high specificity and sensitivity, but the complex interactive networks of cytokines and chemokines in health and disease has made it crucial to evaluate them in relevant groups rather than individually. For example, in some cases, the levels of cytokines and chemokines within a pathway in relation to each other are more important than understanding the individual concentrations. So, multiplex assays have been developed as an alternative to ELISAs for simultaneous measurement of multiple proteins in a single sample. ELISA only allows to measure one analyte at a time. So, if you're planning to use multiple analytes, multiplex assays would be a better choice. Multiplex assays are also a good choice when you have a limited sample volume, and they are also time saving as compared to ELISA and could also be more cost effective. The Abaxis Piccolo Express Clinical Chemistry Analyzer is a clinical chemistry system that can be used for accurate point of care testing. We can offer 11 panels on the Piccolo Express which are clear waved. Some of these include the metabolic panel, the lipid panel, the renal panel and the general chemistry panel. The Eppendorf Master Cycler Pro PCR system is used for traditional PCR, which will be used for applications such as sequencing and genotyping. The Applied Biosystems real-time PCR system is used for sensitive, specific detection and quantification of nucleic acid targets. It is used for applications such as quantitation of gene expression, microarray verification, SNP genotyping and microRNA analysis. The Dionex High Performance Liquid Chromatography. So, HPLC is a form of column chromatography that pumps sample mixture or analyte in a solvent which is known as the mobile phase at high pressure through a column with chromatographic packing material, which is the stationary phase. Each component in the sample interacts differently with the absorbent material, causing different flow rates in the different components, leading to separation of the components as they flow out of the column. The CNR lab has an HPLC equipment which is the Thermo Scientific Dionex Ultimate 3000 system, which provides excellent chromatographic performance while maintaining an easy and reliable operation. The main components include an autosampler, a pump, a solvent rack, and a column compartment, variable wavelength detector, and a software which is used for analysis.